So in this video, I'm going to talk about translation. And what translation is converting mRNA. It's essentially converting mRNA into amino acids slash protein. And this occurs at the ribosome. And mostly at the rough ER. Remember, the rough endoplasmic reticulum has a lot of ribosomes studded all over it. And so let's talk about the whole process. Let me clear this. And so this is a good diagram of what's actually going on in the ribosome. And so translation occurs in 3 steps. One is initiation. 2 is elongation, and 3 is termination. And so briefly, initiation occurs once, it's what gets it started. Elongation occurs over and over, it happens every time you add an amino acid to a chain, and termination happens once when the protein is finished. And so first let's talk about initiation. So let me clear this. And so for initiation, what happens is the mRNA binds to the ribosomes. First, the start codon, which is always start codon, which is always AUG. And sorry if I didn't explain what a codon is before. A codon is a set of three base pairs, and they can be made up of a couple of different letters. For example, in mRNA you have U, A, C, G corresponding to uracil, adenine, cytosine, and guanine. And so you have a bunch of different combinations of that, and that's called a codon. So remember, essentially a triplet. And so as we discussed before, a tRNA has the anticodon, which is a thing that binds to AUG. So in this case, it would be UAC. Remember, A pairs with U and G pairs with C. And so this tRNA, it always has an amino acid attached to the end of it. And so as you can see down here, the ribosomes has an A, P, and E site. And so the E site is where the tRNA, after it gave off its, actually let's start with the A. So the A site is where the tRNA first goes to. And then the ribosome will slowly move up that way. And then eventually the A site will become the P site. And then so what happens at the P site is the P site is where the growing polypeptide chain is. So as you can see, the polypeptide chain is right there, attached to the P site. And then finally, the E site is where the tRNA is released. And so now back to initiation. Initiation is essentially just bonding of the tRNA UAC, UAC to the mRNA. And so the next step is elongation. So let me clear this. Step 2 is elongation. And so what occurs here is the polypeptide, which is a chain of amino acid, grows in length. And so what's happening is each tRNA comes in, and then the ribosome moves further along in this direction and keeps adding amino acids on. And as you can see, you need a specific codon a specific sequence that will bond to the mRNA. And so only one tRNA can bond there, and that tRNA only holds one amino acid. And so when the whole polypeptide is synthesized, there will be no variations. And so remember, peptide bonds they form by dehydration, synthesis. And that's very important. And that's the removal of a water molecule to bond to amino acids or other molecules. And then finally, the third step, let me clear this, is termination. And so what happens is the polypeptide, this whole chain, is released by a release factor. So polypeptide is released by release factor. And so more specifically, it happens when the ribosome reads a stop codon, and the stop codons vary. It could be UAA, UAG, or UGA. As compared to the start codon, there's only one, it's AUG. And these codons, they do not code for an amino acid, so no amino acid. And then so eventually the ribosome releases the mRNA and then dissociates into two subunits. And then the mRNA can be used and read by another ribosome.